Okay, now we want to start this lecture off talking about pleiotrophy, which is the opposite of polygenic inheritance. Remember with polygenic inheritance, we had many genes affecting one characteristic. Now we have one gene affecting many characteristics. One of the things with pleiotrophy, it's often seen in basic structural genes of an organism's body. An example of pleiotrophy is brachydactyly, or the other name for it is short fingers. It's an example that's found in humans. Again, and you'll see that this one gene, having this one gene absent, which is generally, again, it says seen as basic structural genes. So in other words, one of the most abundant body proteins, if you remember, is called what? Go back to your chapter 3. The most abundant body protein, that was collagen. So if there's a issue with this gene, if there's a default, in the gene, a mutation in the gene, it says that you could result in having brachydactyly. Here you could have extra phalanges, absent phalanges, short thumbs, short femurs, for example. But not only does it affect it says it could also affect your other issues with the skeleton, heart problems. So again, we're talking about one gene affecting many different characteristics. Weak arteries, okay? So all because there is a single defect in the collagen gene, okay? And remember we said the collagen is a structural molecule, okay? Collagen produced as fibers. It's a structural molecule, okay? And it says a single defect or mutation in this gene can affect many different characteristics. This gene is generally, if there's a problem with it, it's generally fatal during infancy. Now, one of the things about this is an autosomal dominant disorder. So if you remember from the end of the end of the Mendelian genetics, we talked about disorders, autosomal dominant disorders and autosomal recessive disorders. Well, this is an example of an autosomal dominant disorder. Remember, anytime we say the word autosome, we're saying any other chromosome other than a sex chromosome. So if this is an autosomal dominant disorder, I could ask you on a test, for example, if could two parents produce a child under an autosomal dominant disorder who would not have the disorder? Remember, one of the things I encourage you to do was to work your way back. And I could ask you, well, what are the genotypes of the parent? What's the genotype of the child? You could tell me that the genotype of the child would be what? They don't have it, so we know that they would have to be little b, little b. So the question asks, could two parents produce a child who didn't have it? The answer is yes, because any one big B is gonna give it to mom's sperm, for example, and mom's egg, forgive me, and dad's sperm. So it says, yes, you could get a child. That's who got it here. This child has it. This child has it because it's an autosomal dominant disorder. So any one big B makes them have it. So if we looked at the phenotype, it says the first box is what? They have brachydactyly. Okay, so there's one. Here's another one, brachydactyly. So we ended up with 3 with the brachydactyly, and only one of them, but yes, so the answer is yes, 2 parents could produce an unaffected child. The other question could say, well, what is the percentage of that happening? And you could say who? 1 in 4 is what? 25%. I got a 25% chance of having a normal child without brachydactyly. And they got a 75% chance of having children that do have it. All right, let's keep moving. That was Pleiotrophy. Gene linkage. Now, current estimates place the number of human genes at about 100,000 or more. There's only 23 chromosomes, so that obviously tells us that there have to be many genes on a single chromosome. And if you remember what a chromosome looks like, here is a chromosome that's all wound up. So we're saying that the chromosome doesn't have just one gene location, but it has many different gene locations on that one chromosome. So since there's only 23 pairs, we know that those 23 pairs are fully loaded with more than one gene. All right, so genes occur on the same chromosome, tend to be inherited together. The reason being is because all of these genes that are on the same chromosome says they do what? They move into the gametes together. It's saying that you're not going to rip the chromosome apart and only this part going to go in and then the rest going to go in another one. No. It says whatever genes, they're all linked together because they're all on the same chromosome. So they're said to be linked together. Now let's talk about, it says these genes are said to be linked together and they occur in these what we call linkage groups. Now let's talk about the next, if I can get the slide to go. It says most instances of gene linkage are difficult to determine because for most of us we don't know all of the positions on the genes. Now let's go into sex-linked genes. So that automatically tells us that it's going to have something to do with our sex. All right, the sex chromosomes, it says they also carry genes that are not involved in sex determination. These genes are said to be sex-linked genes because they occur exclusively more often in one sex than they do the other. The X chromosome, as we know, is the largest human chromosome, having thousands of genes on it. The Y chromosome of the sex pair is the smallest, and we know that girls get 2 X's, boys get an X and Y. So basically just the genes for their maleness. Now, 2 disorders that we need to talk about are red-green color blindness and hemophilia. These are examples of some human sex-linked traits. So again, remember when we say human sex-linked, we're saying they occur more frequently in one group of people than the other. So both traits are recessive to the normal alleles for color vision and blood clotting. Both traits occur, again, more frequently in one than they do the other. So where would you predict these genes for the traits to have their loci? Would they be on the X chromosome or the Y chromosome? So red, green, color blindness, and hemophilia. Now, one of the things that we just pointed out was that both traits occur much more frequently in males than they do females. So, again, where would you predict the loci? Is it on the X chromosome or is it on the Y chromosome? Wherefore, to occur more frequently in males, it would have to be on the X chromosome, not the Y, because women don't get a Y. Okay, trying to be silly. All right, so they both have their loci on the X. The reason why men are more susceptible is because if it's on the X chromosome, and again, women don't have a Y, so it would have to definitely be on the X, but men would have more of a problem because women get two X's, so they get an X to negate the problem, whereas men get one X, and if they got a bad X, then they're going to have whatever that issue is. All right, so let's look at some issues with hemophilia. All right, so let's use the following. So we need to figure out what our alleles look like. All right, so X superscript capital H, that's going to lead to normal blood clotting. X lowercase h, superscript lowercase h, that's going to be the hemophilia. And then we will have a Y. And that's going to lead to someone being a male, so the Y chromosome. So for the normal female, we're going to have, and I'm going to pull me another pen, which I could just go ahead and stick with red, but I want to use something different. All right, so two X's, we know that that's going to be a normal female. So we want to stick in the H's here. So X capital H, we know that that's going to be a normal female. X capital H, superscript capital H, and X superscript lowercase h, we know that that's a carrier female. She can pass along to her children, but she doesn't have it. And then the X lowercase h, X lowercase h, that's a hemophiliac female. She's full blown. All right, and then for the men, there's only two possibilities, X capital H and a Y, and an X lowercase h and a Y. Okay, so let's perform a cross. Okay, so here's a sample cross. A man with hemophilia marries a woman who is a carrier of hemophilia. One of the things that I encourage you to do is when you start reading these sentences, is to always try to figure out what the genotypes of the parents are. So it says a man with hemophilia, this tells us that he has to be, said he marries a woman who's a carrier, so that tells us that she has to be, so we got those two, so you always want to figure out who your parents are. It says, well, what is the probability of their sons having hemophilia? What is the probability of their sons having hemophilia? Okay, so we want to do what? Separate and segregate those. And so we end up with dad's gametes and mom's gametes. So then it ends up with a normal male and a hemophiliac male. So we end up, it says, what is the probability of their sons having it? 1-to-1, because one is going to be what? Normal. The other one is going to actually have hemophilia. So a 1-to-1. All right, so what is the probability of their daughters having it? If we go back up here, here's the normal female, and here's the hemophiliac female. So also a 1-to-1 ratio. All right, so here's the okay. Okay, so genetic, let's go with sex linked. Again, genetic, females are red-green colorblind only if both their chromosomes are defective. And whereas genetic males, we know, are colorblind only if their one X chromosome is defective. All right, so let's go to the last one, which is sex influenced. Under sex influence, it's similar to sex linked in that the traits occur more frequently in one sex than they do the other. It differs, however, because the genes that are controlling are no longer sex chromosomes. So we're back to the autosomes. Sex influenced genes are found on non-sex chromosomes, again called the autosomes. All right, the actions of these genes are influenced by the sex of the individual carrying the gene. So in other words, remember before we said environment was important. So environment is going to play a role in the traits that you see. So for example, since we're talking about sex influence, it has something to do with the influence of the hormones. The estrogen, the testosterone, for example. So sex hormones are a part of the environment for all of our genes. All right, so what we want to look at as our example is male pattern baldness. All right, so let's look at the alleles for male pattern baldness. Wrong. All right, the gene for hair on the head has two alleles. Capital H says you're going to have hair on the head. The lowercase h says you're going to be balding. The three genotypes possible are going to come up, but we also have to look at the sex, okay? It's going to depend on whether you're male or female because we said it had something to do with the hormones. All right, so let's look at the genotypes. So the genotypes are a homozygous pair of dominant alleles, heterozygous, and a homozygous pair of recessive alleles. Okay, so it says if you get the homozygous dominant pair of alleles, you have normal hair. For the female, also normal hair. It says if you get the heterozygous pair of alleles, it says you're balding by the time you're 30 if you're a male because of that testosterone we know has to be. And for a female, you're probably still normal. Yep. And then for the homozygous recessive pair of alleles, sounds like doomsday. If you're a male, you're going to be balding by the time you're in your 20s. But for the female, you are not bald until after menopause. And the reason being is because at menopause, you have lost your estrogen, which was negating the effects of testosterone. So you're balding after menopause. So we said female pattern baldness, again, is due to the fact that estrogen was counteracting or negating the balding effect of testosterone. This ends all of our PowerPoints for Unit 4. If you have any questions, please feel free to give me an email or call.